Now, this at first glance is unbelievably complicated, but it's easy to ignore the little boxes and just see these things, which is, at least at this point in time, my intention. So this is just showing different ways that you can specify different monosaccharides. So first of all is this whole idea of carbohydrate. So if you have just a unit that consists of CH2O, that's the basic formula. If you put an N there, you have the different number of carbons that make up an individual carbohydrate. And this is just different ways that you could show this CH2O. Here we have a triose. It consists of 3 carbons. So you have an H and a C and an OH, or CH2O3. This can either exist as an aldehyde sugar, an aldose, with the double bond O on the end, or a ketose with the double bond O in the center. So 2 comments on those structures. The first is that in order to have the number of hydrogens and oxygens and carbons all add up, so you have this CH2ON, so that you have as many carbons as oxygens and twice as many hydrogens, then you simply have to have a double bond O somewhere in there. If you made it into some other kind of structure, then you would have more hydrogens or what have you, and it would no longer be CH2ON. So it kind of falls out of the formula that the double bond O has to be there. And it either can be on an N carbon or a middle carbon, and that actually changes the chemistry. The other thing is that those 2 components are the 2 simplest of sugars, and you're going to see derivatives of those again when we talk about glycolysis, which is part of how you take the molecule glucose, for example, and convert it into ATP. Part of the process is converting the glucose into derivatives of those 2 molecules. So pentoses, 5 carbons. A pentose is important because you see those as the sugars that form the basis of nucleic acids, whether they're RNA or DNA. With DNA, you're actually missing one of the oxygens that's associated with this pentose. So you no longer have CH2O5, it would be actually, however you'd spell it out, minus 1 oxygen. And that's why DNA is called DNA. RNA is ribonucleic acid because it's based on the sugar ribose, and DNA is deoxyribonucleic acid because it's based on deoxyribose, which is missing an oxygen, so deoxy. And here we have a hexose, so 6 carbons. And this hexose is glucose. It's actually called alpha-D-glucose. And you have OHs sticking up and sticking down. What's being drawn here is a representation of a ring structure form where the hydroxyl groups are either found above or below the ring. And so down means below and above means above the ring. And you can't switch these places because in order to do that, you have to actually break that bond and flip them around. And glucose specifically has an OH sitting down, an OH sitting up, an OH sitting down, this carbon plus an OH sticking up. And then to complete the 6-member ring, you don't have a carbon there, you actually have an oxygen there. This hydroxyl group can be down or up. And actually that changes the properties of the glucose in important ways.